1, 2, 3.
Let us pretend for the next 3 minutes that what I have here is the gene that makes you awesome. This gene is just one of a staggering amount of genes in your DNA, but it is uniquely characterized by the specific order of its building blocks, which are called nucleotides. Now, there are 4 different nucleotides, and they always come in pairs, which means a yellow one will always bind to a green one, and a blue one will always bind to an orange one. Let's say science has devoted all of its resources into investigating your gene of awesomeness. Therefore, we, and I'm counting myself as a part of science, are planning an insane amount of experiments, so we need an even more insane amount of your gene. How do we obtain it? And preferably without killing you in the process. Well, we use the polymerase chain reaction. It was invented a little over 30 years ago by Nobel Prize winner Kary Mullis, and it rapidly evolved in one of the key techniques used in almost any research that involves DNA. And it goes a little something like this. After we have extracted your DNA, be it in a lethal or a non-lethal way, we add it to a little cocktail, a scientifically accurate cocktail which has but 3 key ingredients. Obviously, it has the building blocks, the nucleotides I mentioned earlier. Second, if I can get it. Sorry. It contains primers. Primers are a small piece of DNA that will bind very specifically only to the start of your gene. And lastly, we need a little machine. Cells have a number of different machines which are called enzymes, and they're basically just proteins with stuff to do. The enzyme we need is called a polymerase, and after it has located the primer on your gene, it will stitch the nucleotides to the primer, making a duplicate of your gene. After this reaction, we still have ingredients, so we restart the process. We now have 2 copies. Each will bind a primer. Each primer will be elongated, and then we have 4, and then 8, 16, 32, and so on and so on. Now, I can see some of you are already thinking about the possibilities, the endless possibilities, and some of you are thinking, what the hell can we do with this PCR reaction? Well, I'll tell you, I'm a biotechnologist, which means I can run wild and free in the playground that is a biotechnological lab. I use it at my internship to take one gene from one fungal species and insert it into another. Recently, it was also used to take the genome of the wolf and insert it into an Asian elephant in order to get mammoths to roam the earth again. And perhaps, maybe, if I get lucky, one day I discover the gene of awesomeness. And when that day comes, ladies and gentlemen, I will promise you, I will make sure that every single... Thank you.